I'm gonna start with chapter 2, that is sexual reproduction in flowering plants. I'll just read through it and underline the important points and explain it from the textbook itself. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we soon over, the rich colors that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. One important point to note is all flowering plants show sexual reproduction. So whenever you see a flowering plant, just remember that they show sexual reproduction. A look at the diversity of structures of the inflorescences, flowers, and floral parts. Inflorescences means the complete flower parts. Shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, that is, the fruits and seeds. So what is the end product of sexual reproduction in angiosperms? That is, the fruits and seeds. For humans, the end product is baby. But for angiosperms, it is fruits and seeds. In this chapter, let us understand the morphology, that is, the form or the shape, structure, and the processes of sexual reproduction in flowering plants, that is, angiosperms. And now the next we'll move to flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of, just remember, S-C-A-R-O, scaro. Flowers are objects of social, cultural, aesthetic, religious, and ornamental value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of 5 more flowers that are used in social and cultural celebrations in your family. Have you heard of floriculture? What does it refer to? Floriculture refers to the plantation of flowers in large scale. To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. So underline, flowers are the sites of sexual reproduction. In class 11, you've read the various parts of a flower. Figure 2.1 will help you recall the parts of a typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? So next we'll go to 2.2, that is pre-fertilization structures and events. So what do we mean by fertilization? Fertilization means the fusion of male and female gametes to form a zygote, right? So before the fusion of male and female gamete, what happens is called as pre-fertilization. So let's just go through it and study. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated, which lead to the differentiation and further development of the floral primordium. Inflorescences, which means complete flower parts, are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, that is, the androecium and gynoecium, differentiate and develop. You would recollect that the androecium consists of a whorl of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. So what is the reproductive organ of male and female? For male, it is androecium, and for female, it is gynoecium. So we'll learn more about stamen, microsporangium, and pollen grain. So remember, this topic is all about androecium. It's for the male, okay? And stamen is made up of 2 parts, that is filamen and the anther. So just remember, filamen plus anther is equal to stamen. Okay, let's read. Figure 2.2a shows the 2 parts of a typical stamen, the long and slender stalk called the filamen, and the terminal, generally bilobate structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. You see this part, the thalamus or the petals of the flower. So the proximal end of this stamen will be attached to the petals or the thalamus of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen, it's from 10 flowers, it's from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobate with its lobe having two theca, that is, they are having two face, that is, they are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther, figure 2.3a. So when we talk about transverse, just remember that it is the extended version, okay? The bilobate nature of an anther is very distinct in the extended section of the anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in its lobe. So there are four microsporangia, 1, 2, 3, 4. There are four microsporangia and then two in its lobe. The microsporangia develop further and become pollen sacs, and these microsporangia, they will develop to form pollen sacs. So pollen sac means they will act as storage, right? Sac storage. So what do they store? They extend longitudinally all through the length of an anther and are packed with pollen grains. So these pollen sacs, they will store pollen grains. Now we'll go to structure of microsporangium. In a transverse section, that is in an extended section, a typical microsporangium appears near circular in outline. The outline is circular, okay? It is generally surrounded by four wall layers. We'll see it in the next page. Epidermis, endothecium, middle layers, and the tapetum. Okay, four layers. Epidermis, endothecium, middle layers, and the tapetum. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. So the other three wall layers, this three, will help in the release of pollen. And the innermost wall layer is the tapetum and will nourishes the developing pollen grains. See, they all work well together. Epidermis, endothecium, middle layers, they will help to release the pollen, and the pollen will be nourished by the tapetum. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? For me, I think that a tapetum possesses dense cytoplasm because it contains food, right? Since it needs to provide nutrients, it needs to contain food, so the cytoplasm become dense. Okay. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of its microsporangium. What is tissue? Tissue is a group of cells, right? So this group of cells called sporogenous tissue is occupied. They occupy the center of its microsporangium. Since there are four microsporangium, they occupy the center of this all four microsporangium. So now we'll go to microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergoes meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? Ploidy means the number of the cells of the tetrad. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. So what is microsporogenesis? From the name itself we can define it. You believe me? Because The process of genesis means the process of formation, right? The beginning. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. It's very simple. The microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. So at first, the anthers will mature and dehydrate, and then the microspores will dissociate and they will form pollen grains. Inside its microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. And so now we'll get to know more in detail about the pollen grain. The pollen grains represents the male gametophytes. So don't be confused. If the question asks like, what is the male reproductive organ? That is the androcium. But when we talk about the male gametophytes, the gamete, it is pollen grain. If you touch the open anthers of hibiscus or any other flower, you would find deposition of yellowish powdery substance, of yellowish powdery pollen grains on your fingers. Sprinkle these grains on the top of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent, that is important or noticeable, two-layered wall. The outer hard layer, the hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic material known. Exine, external, so exine is the hard outer layer. And this sporopollenin can withstand high temperatures and strong acids and alkali. No enzymes that degrade sporopollenin is so far known. Pollen grain exine has prominent apertures called germ pores where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the intine. So inner, internal, so intine. It is a thin and continuous layer made up of cellulose and pectin. Exine is made up of sporopollenin and intine is made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains 2 cells, that is vegetative cell and generative cell. So this pollen grain have 2 cells, vegetative and generative cell. The vegetative cell is bigger, has abundant food reserve, and a large irregularly shaped nucleus. So how I know is vegetative is bigger, right? So vegetable is a food, right? It's a food and it's present in plenty amount in our place, right? So vegetative cell is bigger, has abundant food reserve, and a large irregular shaped nucleus. Generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are set at these 2 cell states. In the remaining species, the generative cell divides methodically to give rise to the 2 male gametes before pollen grains are set, that is 3 cell states. 2 cell states and 3 cell states is nothing to be confused about, but it's very simple. See, we say that pollen grains have 2 cells, right? So the generative cell and the vegetative cell. If the pollen grains are set at this vegetative cell and generative cell, it is called 2 cell states. Since they contain 2 cells, it is called 2 cell states. But sometimes the generative cells will divide into 2 by mitotic division. Then we will now have 3 cells, right? And if the pollen grains are set at these three cell states that it will be called as three cell states because of it contains three cells. Two cells, that is two cell states, and if it is three cells, it will be three cell states. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous, that is, occurring everywhere in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and racehorses. So let's go back. Pollen grains are rich in nutrients. So what provide nutrients to the pollen grains? We say that the nutrients are provided by the tapetum, right? So the tapetum will provide the nutrients to the pollen grains, and that's why pollen grains are rich in nutrients. And when they are set, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. Viability means how long they are able to successfully deliver the pollen grain into the stigma. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Raceaceae, Leguminosae, and Solanaceae, they maintain viability for months. You may have heard of storing semen or sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen, minus 196 degrees Celsius. Don't forget, minus 196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs. So now we're done with the male reproductive organ, and now we'll go to the female reproductive organ, that is the pistil, megasporangium, that is the ovule and embryo sac. The gynoecium represents the male reproductive part of the flower. For the male, it is the endoecium, right? The gynoecium may consist of a single pistil, that is monocarpellary. Monocarpellary means single pistil or may have more than one pistil. So what is pistil? Pistil is stigma, style, ovary together will constitute the pistil. Okay? And if they have only one pistil, they will be called monocarpellary, and if they have more than one pistil, they will be called as multicarpellary. When they are more than one, the pistils may be fused together, they are called as syncarpous, or may be free, that is apocarpous. Its pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated, slender part beneath the stigma. The basal bulbous part of the pistil is the ovary. Inside the ovary is the ovarian cavity, that is the locule, and the placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you've studied in class 11. So as you can see from this diagram, the stigma, style, and the ovary, these three constitute the pistil. Arising from the placenta are the megasporangia. So, as I've said, microsporangia is for the male and megasporangia is for the female. Commonly called ovules. The number of ovules in an ovary may be 1, that is wheat, paddy, mango. So, just remember, ovules will form the seeds. How I know is, see, there will be ovary and the ovule, right? If you see E here, right? So it will be seed, and ovary will be the fruit. If you see E here, then it is seed. E, right? E. Okay. it's simple. The number of ovules in an ovary may be 1. The number of seed in a fruit, okay, may be 1. Wheat, paddy, mango. Have you ever seen mango having 2 seeds? I don't think so, right? They have only one. They have only 1 seed. And too many, papaya, watermelon, orchids. So the megasporangium or the ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. So this is the ovule, okay? See, in here it's the ovary here, right? But this is the, the enlarged portion. Okay, so now we'll learn more about the megasporangium, that is the ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule, which is shown in the figure 2.7d. The ovule is a small structure attached to the placenta by a stalk called funicle. Okay, this stalk is called the funicle. And the body of the ovule fuses with funicle in a region called hilum. Okay, this part is the hilum. And the funicle, it will be fusing with the funicle, and that is called the hilum. Thus, hilum represents the junction between ovule and funicle. Its ovule has 1 or 2 protective envelopes called integuments. See, outer integuments and inner integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. So there will be as we proceed, there will be a reason why this part why the micropyle region is open. Okay. Opposite the micropylar end is the chalaja, representing the basal part of the ovule. Enclosed within the integuments is the mass of cells called the nucellus. This part is the nucellus. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte, and the female gametophyte or the embryo sac is located in this place, that is the nucellus. Okay, this is the embryo sac. So now we'll move an ovule generally has a single embryo sac from megaspore. Alright, see, the ovule generally has only 1 single embryo sac. Afterwards, we'll learn about the differences between microsporogenesis and megasporogenesis. So now we'll get to learn more about the megasporogenesis. The process of formation of megaspores from megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergoes meiotic division. What is the importance of the megaspore mother cell undergoing meiosis? Meiosis results in the production of 4 megaspores. So now let's try to differentiate between microsporogenesis and megasporogenesis. So microsporogenesis, this is the definition itself: the formation of microspore from pollen mother cell by meiosis. And megasporogenesis is the formation of megaspore mother cell by meiosis. And microsporogenesis, as we said, it is for the male, right? It takes place in anther. And for megasporogenesis, it's for the female, so it takes place in ovule of ovary. And microsporogenesis, it will have 4 functional microspores are produced. Okay, all the microspores will be functional, but megasporogenesis, they have only 1 functional megaspore. That is, only 1 functional megaspore is produced, so we have only 1 embryo sac. Okay, these are the difference.